Everyone can see the screen that I'm sharing, people?
Yes, sir.
Yes, sir.
Okay, perfect. So who would like to go with a quick recap of the last class? Someone raised hand. Great. Parker raised hand. I think this is the first instant where someone raised hand for this, and I'm really excited. Great, Parker, go ahead.
Yes, sir. Can you hear me?
Yes, I can hear you. You raised hand for the summary, right?
Yes, sir.
Perfect. Go ahead.
Yes, sir. So we studied about the reproductive organ of angiosperms, which is a flower, and flower is
Angiosperms are
also
flowering
the
plants.
modified
You're
angiosperms of
right.
flowering plants.
Yep.
We studied about how ovules are future seeds and ovary is our future fruits.
Right.
We also studied about a true fruit and a false fruit. A true fruit is only produced from a ripened ovary, and no other tissue is involved for the ripening part. And the function of a flower is to carry out pollination. And petals attract pollinators. Then we studied about pre-fertilization events. In pre-fertilization events, the development of the flower takes place. Then we studied about the parts of the flower and
Yes. What was special about parts? Anything you remember? How a flower is structured layer by layer?
the first layer, which is in the innermost layer, which is the pistil. Then,
Right. Which is the female
then
reproductive
comes the
part, right?
stamen. Yes, sir.
Yeah.
Then comes the stamen. Then comes the petals or corolla. Then comes the sepals or calyx.
Right. And we also discussed the reasons for which they evolved, the potential function that they serve in a flower. So sepals or calyx is mostly for protection. Petals are the attracting agents, like they attract insects on the basis of vibrant colors and also sometimes the scent that they produce. And they also allow, petals also allow, when the insects approach the flower, they also allow that landing, the kind of a helipad, the landing site where the flower, where the insect like honeybee lands. Okay, cool. Then anthers, you know that they are, stamens, you know that they are the male reproductive part. It consists of anther and filament, and pistil is the female reproductive part, which consists of three parts: stigma, style, and ovary. We will come to the details of these in a bit in today's class. But yeah, that's what it was. And anything else, Parker, you would like to say?
We studied about the
Mhm. How the androecium and gynoecium develops, right? So this comes, so we can broadly categorize the whole process of sexual reproduction in plants, flowering plants, into three categories. One is pre-fertilization, what happens before the fertilization takes place. It's basically structure development, attracting pollinators, and pollination. These three things in this series, in this order. Then fertilization includes formation of pollen tube and fusion of the male and the female nucleus. And post-fertilization is the result of this, that we see that petals and sepals are shed off and ovary gets ripened to form fruit and ovules develop to form seeds. I think we started with the first category, pre-fertilization events. where we talked about development, right, Parker?
Yes, sir.
What did we discuss? Where did we left? I think this, yeah.
We were studying about the tapetum.
Tapetum, yeah. So I told you that we first started with the. I think yes. Yes, Iram.
So I'm so sorry to interrupt. Says the spotlight feature.
Yes, it's on. Yep. Yeah. So I also told you about how the events of development takes place, starting from the development of floral primordium. So the name suggests floral means flower, primordium means something primitive or before flower. So before flower, the tissue that develops is called floral primordium. This gives rise to the floral buds, and then the buds become flowers. And within the flower, you can have androecium or gynoecium or both. So androecium, um, which is also known as the stamen, is the male reproductive tissue, and gynoecium is associated with the female reproductive tissue, which is pistil in the flowers. Great to see you, Iram. How are you doing?
Good, sir. How are you?
Good. Okay, so and the second thing I told you, we went into the structure of microsporangium, which is the pollen. I told you that micro word is always associated with the male gametes. So in a stamen that's divided into anther and filament as structure, the anther is bilobed in most of the angiosperms, and in each lobe there are two compartments present, like called theca. So it's called a bilobed diathecous arrangement. Every lobe is diathecous, and the structure of the microsporangium, if you go within the layers of this lobe, then the outermost is called the epidermis. And as you know that all epidermis play the role of protection, even our skin, the epidermis of this, it's called the epidermal layer. It serves as protective agent. So it's for protection, then endothecium, middle layers, and then the tapetum. Tapetum is the innermost layer, which is in actual contact with these developing pollen grains, these cells, which are the microspores. So tapetum provide nutrition to microspores. That's why they have a dense cytoplasm and they are multinucleated. By being, now you have studied genetics, so let me try to connect a dot here. Let's see who gets closer to the answer or who gives the right answer. What is the benefit of being multinucleated for tapetum? That's a legit question which is asked in boards, and a direct answer is not addressed in this manner in the NCERT, but there is the answer. What do you think is the benefit of having dense cytoplasm and multiple nucleus for tapetum? Anyone? Quickly write down the question. Why is tapetum multinucleated with a dense cytoplasm? Why is tapetum multinucleated with a dense cytoplasm? Yeah. Anyone would like to take a guess? So if you take a guess based on logic, most probably that guess will be either correct answer or very close to correct answer. No one? What is the function of tapetum? Come on, quickly. Parker says it provides nourishment. That I only told you. Yes, Mariam, the question is, why is tapetum multinucleated and has dense cytoplasm? So Parker says it provides nourishment to pollen grains, but what is the reasoning behind? It could not have provided nourishment if it was uninucleated and had a normal cytoplasm. Parker? So when you give answers, you also have to give the logic behind that answer. You're right that it's related to, because its function is to provide nourishment. But can you connect the dots from genetics chapter? Transcription, translation. What happens if a cell has multiple nucleus? If it's multinucleated, what happens? What does it mean? Yes, Iram. You're mute, I guess.
Yeah. So it is. So it's seen in this condition is seen in coconut, where the endosperm. I'm not getting the word for it.
Right, but try this. You're right. So let's break down this problem. Let's give simple answers. What happens when a cell is multinucleated? What happens when it has multiple nucleus? Can we say that if it is multinucleated, then it will have multiple copies of the same chromosome? If it has 4 nucleus, it will have 4 copies. If it's and in this case, because these will be diploid plants, so it will have 8 copies now, 8 copies of the same gene. And if that gene is produces a protein which is a nutritional protein, then you can make 8 times that protein because you have 4 nucleus in each cell. Does it make sense? More nucleus means more information, more genetic information available. You can do more transcriptions, more translation. You can produce more proteins and other substances within the cytoplasm. And that's why the cytoplasm becomes denser. Okay, because there are multiple nucleus. All of these nucleus are active. They are transcribing, and those mRNA are coming out into the cytoplasm. Those are getting translated by ribosomes, and a lot of protein is in the cytoplasm. So if a lot of protein is in the cytoplasm will be dense, right? And that's why these cells are very good nourishing cells, because they have abundant of everything, more than, so they can fulfill their own requirements, and they can also fulfill the requirements of the cells nearby by providing them with the proteins. Does that make sense, everyone? So that's why tapetum is multinucleated. Okay, now there is something called a sporogenous tissue. So the sporogenous tissue, when these anthers are very young, when they are developing, right at the center, there are a group of very, very tightly packed cells, which are called sporogenous tissue. Okay, now these sporogenous tissues basically develop into spores, microspores. Okay, so today we'll start from here. Let's start with the process of microsporogenesis, like we decided. And I told you to go back and read human reproduction, right? Revise human reproduction to discuss a parallel, to draw parallels, which means similarities and dissimilarities between the two. So when I say microsporogenesis, does something rings in your head, something similar? Spermatogenesis. There also we were making male gametes, but in an animal system, which is human. Here we are making male gametes, but in a plant system, right? So here we call them microsporogenesis. There we used to call it gametogenesis, and specifically spermatogenesis. Here we are making microspores. So first of all, this journey starts with sporogenous cells. Much like, if you remember, I'm not going to draw all the parallels, but I'm telling you how to learn biology so that it becomes very easy for you. Inside the human testes, if you remember, there were cells called spermatogonium cells, right? Spermatogonium cells that divide and give rise to primary spermatocyte and then secondary spermatocyte, and then spermatids, and that develop into a fully functional sperm and comes out of the testes. Okay. Similarly, here we have sporogenous cells, and these sporogenous cells are diploid. But microspores are gametes, right? Gametes cannot be diploid. So gametes have to be haploid. So what will happen? These sporogenous cells will undergo what?
Yes.
Perfect. The sporogenous cells will undergo meiotic division or meiosis, right? Meiosis takes place, and as a result of meiosis, it forms microspore tetrads. Tetra means 4. So group of 4 cells, and that's what meiosis does, right? It takes one diploid cell, and when meiosis completes, as a result, we get 4 haploid daughter cells. Correct, everyone? Make sense?
Yes,
Yeah.
sir.
So we have a microspore tetrad where it's a bunch of 4 cells that looks like this. That's why we call it tetrad. And each cell is a haploid cell. So N, okay? So you can make something like this. This is what a tetrad looks like. Now this microspore tetrad, you know the ploidy I told you, it will be N. Now each of this tetrad is capable of giving rise to a microspore, which means a pollen grain. Okay, so you can write down that so one sporogenous cell, let's do some mathematics because these kind of question come. So if I start with one sporogenous cell, okay, or one sporogenous tissue, it will give rise to one tetrad, which will have 4 cells. Each tetrad will become a pollen grain. So each tetrad, each cell here of this 4 cluster of 4 is a potential pollen grain. Okay, so how many pollen grains can be formed? Come on. Yes, Iram, tell me.
Can you please repeat what you said?
Yeah, I'll repeat the question. So we are doing a little bit of mathematics. So if I start with one sporogenous tissue or one sporogenous cluster, after meiosis, it will form a microspore tetrad, right? Tetrad means there'll be 4 cells. In one microspore tetrads, how many pollen grains can be formed? 4, right?
Yeah.
Because each cell from one microspore tetrad, you can make 4 pollens. Does that make sense? But Mariam was right. Mariam is writing 4. So each of this cell in a microspore tetrad, write down. Each cell of a microspore tetrad is a potential pollen. We call them pollen grains because they are tiny and look like grains under the microscope, but the name is just pollen. It's a potential pollen and is also called as a microspore mother cell, MMC. But you shouldn't remember it as MMC. You should remember it as PMC because we call it pollen mother cell. Because remember, the problem is for the female also we use the word macro. So there we call it MMC. Macrospore mother cell is for the female, and microspore mother cell, we don't use that term. We use pollen mother cell, PMC. So there are 2 distinctions, PMC and MMC. So write down. It's also called PMC, pollen mother cell. So you understood sporogenous cell, meiosis, form tetrads. Within the tetrads, there are, each one is a PMC, right? Pollen mother cell. It will develop to give rise to a pollen grain. Yes, Parker, I'll repeat for you. Sorry, I lost the electricity, but it will be back in some time. So I was saying, Parker, that one sporogenous tissue or one sporogenous cell after meiosis forms a microspore tetrad. Tetrad simply means a group of 4 cells, right? And in this tetrad, each cell is haploid and each cell is a potential pollen. So it can develop to become pollen cell. That's why until it is not developed, it is called a PMC, pollen mother cell. It will become a pollen. Is it clear?
Yes, sir.
Yeah. And now, since you know these terms now, what is a microspore tetrad, what is a sporogenous cell, what is a PMC, let's define microsporogenesis. So write down the definition. The process of producing microspores or pollens, process of producing microspores or pollens from a pollen mother cell. From a pollen mother cell, PMC. Is called microsporogenesis. Okay. And simply what happens, they are clustered to begin with. So I'm just making a boundary around them. They are clustered. And then later on, they're all separated from each other, right? And each one develops into a microspore. So if you open an anther, you will see thousands and thousands of microspores or pollen grains ready and stored inside. Okay, because thousands of these sporogenous cells will undergo meiosis, will produce microspore tetrads, which are potential pollen. So they are called pollen mother cells. These pollen mother cells take nutrition from tapetum, the cells of tapetum, develop into microspores and get stored there in the theca, right? And all it needs now is a dehiscence. So they are all tightly packed inside the anther. So these are all pollens. And how will these pollens come out? So let's say this is in a bilobed anther. Okay, they are filled here. Now the anther needs to just burst, and it bursts and all these pollens come out in the air, right? And if the mode of pollination for that particular flower is insect, it will stick to the insect's body and get pollination transfer will be done and pollination will happen. If it is air, then these pollen grains, if the plant depends on air for its pollination, needs to be very lightweight so that air can just take it with it, right? But if they rely on insects, what should be the property of these pollen grains? Tell me. Logically, it should be sticky, right? So that it can stick to the insect's body, the legs, appendages, fur, etc., the fine hairs that are present on the insect's body. Yeah, correct. So you already know this. Parker is correct. And this is how from the production of microspores happen, and through dehiscence it comes out. Now talking about pollen grain itself, let's go into the detail of pollen grain. So these things are where they ask you the question. So let's say talk about microspore or pollen grain. Just like we said about sperms, thankfully you don't have to go into a very detailed analysis of microsporogenesis, but you should know the basics. So pollen grain. Now, pollen grain represents the male gametophyte, right? The main gamete structure of the plant. Now, if you have you ever seen these pollen grains of any flower? Any one of you has touched the pollen grain of a hibiscus maybe, or sunflower, or any other flower with prominent anthers? Anyone has touched?
Yes, sir. We've done an experiment.
You did an experiment.
Mhm.
That's great. So what did you do? You just brushed it under on a cover slide
Yes, on the cover slide.
and
Yes.
observed it under the microscope, right? So
Yes.
what did you see under the microscope? That's great. What did you see under the microscope? Can you just explain it in words? What was it like?
So I couldn't see.
You
I couldn't see
you couldn't
anything.
see, you couldn't see anything? You are you sure you were zooming properly? Because these are
No, sir.
one of the
Actually, you know
pollens.
what? They don't teach us how to use the microscope, so it was so tough for me to like figure out what is happening. So,
Yeah, that's one caveat in teaching in general. I believe, like, teaching is a very interdisciplinary process. So when I teach students, sadly, I'm not teaching you people offline, but when I used to teach offline, I always used to bring real stuff to the class. I remember when I used to teach offline, if we are talking about the insect larvae, I've even brought larvae to the class and showed students that this is an insect larvae. If it's about a plant, but the point is, Iram is very right. All these things which are in book, seeing is believing in science, right? Even seeing the data for things you cannot see, like air. So we say that we can't see air, right? So how to believe that there is an air? There are many other invisible things like radiations in the solar system or in the universe which we can't see. But how do we believe that they are there? But still, when we do some experiment, we get some data, whether it's spike or any kind of a data which we see with our eyes and say that, oh, this can only happen if there is an air or if there is this invisible thing, right? So seeing is believing. So did you go back? I think you should go back and ask them to teach you how to use microscope first, so that you can see pollen grains for yourself. So pollen grains comes in all shapes and sizes. Okay, like it could be something like a dehydrated resin. It could be something like a very fine. You'll see a smaller, tiny version of a wheat grain, or it could be something like a spiky ball where you will have spikes coming out of it a little bit. They could be tiny, tiny dots. It could be long. So lots of shape pollen grains have. But the common thing in every microspore is that there is a range of their size. So somewhere around 20 to 50, 60 micrometers in diameter are most of the pollen grains. Now, I'm not including exceptions. The smallest and the biggest might be out of this range, but in general, I'm saying. And the most important thing that is there is that it has an outer layer. Every pollen grain has an outer layer which is very hard. Hard in the sense like not that you cannot break it. Physically, you might be able to break a pollen, but chemically you can't. Okay. When I say hard, I say that it's very, very chemically stable, and it is called exyne. So the hard outer layer is called exyne. And this exyne is made up of a substance called sporopollenin. Now this sporopollenin is the organic polymer. Okay. You can write down it's an organic molecule, it's organic polymer. And it is the most resistant, chemically resistant substance, one of the most chemically resistant substance in nature. So you cannot degrade Exyne by any enzymatic activity in our body. So if I eat pollen, I won't be able to degrade or metabolize this sporopollen. So no enzyme in my body, whether it's for protein digestion, amylase, protease, lipase, anything won't work on sporopollen. Okay, do you understand that? Write down, it is one of the most resistant organic compound present on Earth. Now, this is very interesting that plants chose sporopollen to cover the pollen grain. Now, there's a problem. You do want transfer of nucleus to happen from a pollen grain to the stigma. But if you use Exyne to make the outer covering, how do you think nothing can break it? So how do you think nucleus is going to come out of the pollen and go inside the pistil and fertilize the egg cell there? Any idea? I think this is also an experiment. If I remember, in my days in school, we used to get a flower, probably hibiscus, and cut the female stigma and style. Didn't need to take the ovary, just the stigma and style part. Press it under the slide cover slip and observe it under the microscope. And we used to see small tubes growing from stigma to style. Those are called pollen tubes. But how do you think a pollen tube comes out of the Exyne? A simple answer is, do not cover the Exyne with fully around the pollen. So then Exyne has some holes. So it will have holes at different places. But these holes won't be very big, very small holes. And within the Exyne lies the just 1 second, people. Yeah. The inner layer. This is what the cell, pollen cell is like. This is called intine. Exyne and intine. Okay. Now, within every pollen grain, there are two kind of cells. So there are two cells present in one. One cell, bigger one. The other cell is a little smaller. And they have their own nucleus. Okay. Now, these cells are vegetative cell, and anyone knows the other one? Is this chapter done in your schools?
Generator. Yes, sir.
Yeah, right. So vegetative and generative. Which one is which?
The big one is vegetative.
Yes. So the bigger one is vegetative, and it means, so it's vegetative cell. So the nucleus of that cell will be called a vegetative nucleus. And vegetative cell provides nourishment to the generative cell. Okay. Now you will understand later why there is this kind of arrangement in plants. So generative cell, this nucleus is called generative nucleus then. Okay. So you can write down the vegetative cell provides nutrition and helps the generative nucleus during pollen tube, or leads the instead of helps, right, leads the generative nucleus during pollen tube formation. Okay. Now, pollen is one thing, because since we cannot metabolize it. It's something that humans do not recognize. Okay, Parker says, can I repeat the sentence? Iram, would you like to repeat the sentence, please?
Vegetative cell
Yeah, vegetative
uh,
cell
leaves during pollen tube
lead,
formation.
leads the generative cell. Oh, sorry, vegetative nucleus leads the generative nucleus during the pollen tube formation. I'll show you with the diagram how it happens. But since pollen grains are not easy to, I should not say easy, it's almost impossible to metabolize it with the enzymes that we have in our body. So pollen always is identified as a foreign antigen by our body. And it's very, very common these days for people to have pollen allergies. Okay, sometimes it's very specific and sometimes it's general. Any sort of pollen goes inside your body and your immune system is hyperactive to see that substance because it doesn't know what to do with it. Okay, so pollen allergies can happen because of many species of plants pollen, and it causes bronchial infections or inflammations. Okay, it can also lead to chronic respiratory disorders like bronchitis, which is itis means inflammation of the bronchioles, and it can also make situation worse for asthma patients. The patients suffering from asthma, it gets worse. They get an attack the moment if they have a pollen allergy. It's a bad situation. So never take an asthmatic patient who has any sort of pollen allergy to walk in a park where there are lots of flowers. Okay, you might be thinking that, I'm doing a good thing to the person, let's go for a walk in the park with lots of beautiful flowers, but that might not be a beautiful scenario for them because right, there are many plants that have invaded ecological spaces. We call them as invasive species, so, or contaminating species. So back in before the 60s, India was not independent in terms of producing its own grains, whether it's wheat or rice. So we used to heavily import wheat from US. Okay, and with the wheat also came a weed, a grass. It's not basically a grass, but it's a species called carrot grass. The leaves of this plant are very, very tiny and looks like a miniature carrot plant, but it's when you will uproot it, no carrot will come out. It's just a weed, and it's very invasive. In today, I think almost every public park or garden which has open pollination in India, you will find a parthenium. It's called parthenium, the carrot grass. So you will find carrot grass everywhere, okay, wherever there is grasses or lawns and parks. So it's, and the pollen grains of that species is a big allergen and causes allergies in many humans who are allergic to pollen grains. Do you know anyone who's allergic to pollen grains? Anyone knows anyone who's allergic to pollen grains? No. Okay, that's quite good. Okay, so this is a pollen with the generative cell, vegetative cell, exyne, and intine. Now something happens to this. Now let me draw a so this is stigma, and this is style going in, and then you know that it forms a bulging structure with a, this is called ovary, And this is the ovule sac, and there will be present ovules, right? So when this pollen comes and lands, let's say on the stigma, okay? In the open, there are many plants around us, right? Grown. So let's say you go to a garden, you will see rose, whatnot, all sorts of flowering plants. Insects also go from plant to plant. They can even go from one species to another species, right? So there are high chances, and it happens every time, that very different kinds of pollen grains will fall on the same stigma, right? So this is species A, this is species B, and this is species C. So 3 different pollen grains landed on the stigma of a plant. And how will the plant know that which pollen grain should be allowed to form a pollen tube? Because this grain will start forming a tube which will go all the way down through style into the ovary. And through that tube, through that tunnel, to that pipe, these nucleus will travel and go and fuse there. So this is how sexual reproduction happens in plants. So can you take some hint from the animal reproduction where the sperm comes? Remember, we had a discussion. How does ovum recognize its own species sperm? What if, so if I inject the sperm of a different species, let's say of a tiger, into the body of another species, which is let's say rabbit, okay, will fertilization happen? Will we get something which looks like cute like a rabbit but roars like a tiger, is carnivorous like a tiger but white like a rabbit, something? Will we get? No, right? Parker says no. So what prevents it? If I try to make a human sperm fertilize a zebra ovum, will it happen? We'll not get a human baby with zebra stripes. We'll never get a human baby with a zebra stripe, right? Why? Because remember, there was a species specific interaction that used to happen between ovum and the sperm. The moment sperm touches the ovum, there's a series of chemical interactions that tell the ovum that, okay, this sperm is of my species and it's allowed to come, right? Yes, Alia says chemicals. That's no one in the world can say it's wrong answer, but Alia will not get a single mark for writing chemicals, though it's the right answer. Then she writes signal. Great. Now you may get half mark. So yes, there's a chemical interaction that happens. In this case, when any pollen comes and lands on the stigma, the first thing that happens is pollen pistil interaction. Okay, they had a talk before opening the gates. Stigma talks to all these pollens, ask, who are you? Why are you here? Tell me the code word. And if the pollen tells the right code word, which is the right chemicals, then only that pollen will be allowed to produce a pollen tube. All other pollens, let's say in this case, B is the right one. And these interactions are nothing but sugar molecules and receptor which acts as receptors on the surface. So stigma will have, the surface of stigma will have some receptors, and the surface of pollen will also have some receptors. They will interact with each other, and when they will interact, there will be a binding of the ligand receptor, and you will know that, oh, this is the right match. So write down about pollen pistil interaction. We'll first just do pollen pistil interaction, and then we'll move forward. This actually comes in fertilization, but I'm just I'm just teaching you here since we started this conversation. So write down. In pollen pistil interaction, a chemical interaction takes place between the chemicals produced by the pollen with the receptors present on the cell membrane of stigma. With the receptors present on the cell membrane of stigma. Okay. And once this interaction is done, then write down. This interaction helps the pistil. This interaction helps the pistil to identify the right pollen, belonging to the same species, and allows the formation of a pollen tube. Okay. And just, you know this already, these pollen tubes can only come out of these holes where the exyne is absent. Okay. Cool. So this one got selected, the others got rejected, and now it will start producing a tube which will go inside, but it will not enter from the top. It never enters the sac from the top. Okay, it goes all the way around and always enters from the bottom. Okay, why? We'll come to know later. But is that clear? The interaction is clear, everyone? Perfect. What I wanted to say. Okay, now having said that, just before this happens, there is something that happens to pollen. Okay, we'll come to what happens in pollen tubes, but something that happens to pollen. And what happens is the generative cell, which is the smaller cell, it's like it starts dividing. Okay, and just before pollen grains are shed from the plant. Okay, so this what I was telling you is how pollen grain looks when it is inside the anthers. Just before the anther is about to dehiscence and the pollen grains are about to come out, this generative cell divides into two. Okay, sorry, the nucleus. The generative nucleus also divides into two. So, and this is a mitotic division. So write this down. And by the way, sorry, I forgot to tell you this thing. These small holes where the exyne is absent is called germ pore. This is germ pore is from where the pollen tubes comes out. Now write down. In most of the angiosperms, pollen grains are shed pollen grains are shed at two-celled stage, what we have drawn on the board. Okay, are shed at two-celled stage. One, a bigger vegetative cell, and the smaller generative cell. Okay. But in many species of angiosperm, so when I say most, I think ratio-wise, I'm not sure if it has been characterized for all the angiosperms, but my educated guess is that it will be somewhere around 60 to 65% shed their pollens at a two-celled stage. Rest 35 to 40%, if I'm not wrong, shed the pollen at a three-celled stage. So you can write down many species shed the pollen at a three-celled stage. Now, what do I mean by three-celled stage is that this generative cell divides, but this division is mitotic division, sorry. The generative cells divides mitotically to form two male gametes or two male nucleus. Okay, so this is a two-celled stage, which let me write it down here. Two-celled pollen. Okay, two-celled stage pollen. But there's also a condition where it will be like, okay, so there'll be one bigger vegetative nucleus, and the smaller generative nucleus will divide into two. Okay, this is a three-celled stage. So this is the vegetative cell, and these two are two male nucleus. Why it is important to know? Just draw it and write around 40%, 35 to 40%, and this happens in around 60% of the angiosperms. But let me tell you, even in those 60% where it is shed at a two-celled stage, still before the pollen tube starts, the generative cell divides into two. Okay, so for almost all the angiosperms, there are two male nucleus and one vegetative nucleus in a mature pollen. So write down. Regardless of the stage at which pollens are shed, regardless of the stage at which pollens are shed, almost all angiosperms have two male nucleus and one vegetative nucleus in a fully mature pollen. Yes, Iram.
Can you repeat this?
Yes, sorry. Regardless of the stage at which pollens are shed, almost all angiosperms have two male nucleus, and one vegetative nucleus in a mature pollen which is ready to form a pollen tube. which is ready to form a pollen tube. Any doubts till now? If they ask you how many cells are there in mature pollen, you will say 2, vegetative cell and generative cell. Okay, but in many species, the generative cell divides mitotically to give rise to 2 male gametes. Okay. Now remember that there are 3 nucleus now. So if I draw the same thing in a little bit zoomed in manner here, and let's say this is the pollen and it's forming a pollen tube. So you can see the tube now being formed. Okay, this tube is going. Okay. Stigma, style. Now there will be 3 nucleus traveling. 1, and I'll use another color. Let me use green. 2 and 3. Okay. The nucleus that leads is the vegetative nucleus. This order is important. And the 2 nucleus that follow, the 2 nucleus that follow are male nucleus. These are basically the gamete. Okay. Is it clear, everyone? Now we'll stop this here and we'll come back to it later on when we will be actually doing the fertilization, like theoretically, in the plants. Anyone has any doubts still here? If everything is okay, give me a thumbs up, everyone. If everything is clear to everyone. Rabia, Alia has also joined. Mariam, Parker. Perfect. Now, let's go and talk a little bit about megasporangium. So microsporangium is clear. Let's go and talk about megasporangium. Sorry for keeping it a suspense and leaving you on a cliffhanger with this pollen tube going in. We will come back to it. Okay. Now the word megasporangium, what does it mean? Simply ovules, right? So megasporangium is ovule. Now in one anther there can be thousands and thousands of pollen. Okay. But in plants, just like in humans, remember, again, we are drawing some parallels. How many sperms can be present per ejaculation in a human? Somewhere it starts from 30 million, right? 30 million to 300 million of sperms. But how many ovum are there in humans? Normally, 1 single. And in case of non-identical twins, there are 2, right? But let's say 1, or in rare cases, 2, generally 1. In case of other higher organisms like cats and dogs, there can be multiple at the same time. So that's where you get a litter, like a bunch of puppies or kittens being born at the same time. Okay. Another interesting thing is in dogs and cats, when the puppies are born, very interestingly, not all of them will not have the same biological age. So they all might not have fertilized on the same day. Okay. Because the female does not know that she has conceived. There is no menstrual cycle in these organisms, right? Menstrual cycle is only present in primates and human, like primates basically. In non-primates, cats, dogs, goats, cows, buffaloes, there's no menstrual cycle. You will not, anyone who has a pet cat or dog knows, a female pet cat or dog knows that they don't menstruate like humans, okay? There's no bleeding, but they still have a cycle that's called estrus cycle. So the thing is, if a female dog produced, let's say, three ovums, and she's a stray female dog, and she mated with a male dog, and the 3 ovums got fertilized, so there are 3 implantations happened. The next day or the day after, she can produce 3 more, or even next month she can produce 3-4 more. Those can get fertilized and implantation can happen. So if she gives birth to 6 puppies, maybe all of them are not same age when they are born, okay? But because the reason why they are born at the same time is because the fetal ejection reflex is kind of, when she starts giving birth, it's not like she can hold one inside and give birth to 3 or 4. Yeah, just give me 1 second, people. I'll take 20 minutes. Okay. Yeah, so coming back, in plants also, though there can be multiple ovules present in one single ovary, it will always be much less than one single anther producing pollens. Okay, so in some plants, very interestingly, per ovary there is just one single ovule. And how can we say, how can we see and say with proof that this is a plant where in per ovary there is only one ovule? Run your brains. What does ovary becomes? The future fruit. What do ovules become? The future seeds. So if I eat a fruit and I only get a single seed from it and it's a true fruit, can I say that in this plant per ovary there is just one ovule, like mango? So when you eat mango, from each fruit just comes one seed. Avocado, from each fruit just comes one seed out, right? Peach, single seed comes out. Rice, wheat. So from each of this ovary, one single seed comes out in isolation, right? So wheat, rice, mango, they are all mono ovule, mono is a term for it. It's on my, I'll tell you when it comes. But it simply means single ovule is there, okay? That's what I mean to say. So there's a single ovule per ovary. That's why when it became a fruit, the ovary became a fruit, there was a single seed inside it. So that's one. There are also fruits where you open it and you find multiple seeds, like papaya, watermelon. Okay. So when we cut these from one, because that fruit is ovary, so when we cut these from one fruit comes out lots and lots of seeds. Okay. So in that case, we call it multi-ovulated ovary. Yeah, mono-ovulated ovary and multi-ovulated ovary. Is it clear? Yeah. So write down. Mono-ovulated ovaries are mostly, just write down examples: wheat, rice, mango. Multi-ovulated ovaries are papaya, watermelon, what else? All of the fruits, muskmelon, all these melons, and what? Tomato, brinjal, all these, capsicum, chilies, they all have multiple seeds inside. Now about megasporangium, what do we already know about it? Yeah, someone has a question in the chat. Oh no, seeds. Parker says seeds. Okay, so write down. So ovule is a very interesting structure, and also how should I put it? So just write down first. A typical angiosperm ovule, is attached to see, because in plants, one difference between humans, animals and plants is in humans, both the gametes were traveling, right? Sperm was traveling from outside into the female tract, and the ovum was traveling from the ovary in through the fallopian tube into the uterus. Here, that's not happening. Here, the ovule is fixed in the ovary, and it is the male gamete that has to travel all the way through the stigma style and come and penetrate the ovary and go inside and fuse with the ovule. So here the ovule can stay stationary. So it is attached to a structure which in plants also is known as placenta. In humans, when does placenta comes? Placenta comes when fertilization has happened. And after that, it's embedding. And the first tissue that is formed is placenta. The function is same there, because there the baby has to develop, or the progeny has to develop inside the uterus. Here, the seeds develop to the ovules develop to become seeds in the ovary itself. So write down, a typical angiosperm ovule is attached to the placenta, in the ovary, through a stalk-like structure, called funicle, F-U-N-I-C-L-E. Okay, so let's draw this. So this is funicle, and this here will be, this continuous tissue will be placenta. Funicle will attach to placenta, and then this whole structure is something like this. So after funicle, we see a region of attachment, something like this. So it has multiple layers. Let's draw this. And in the center will be your embryo sac. So let me start from the top. So this is funicle, okay, which attaches this to placenta, and this is again, it is in the part of the ovary. So this is the ovarian wall, okay, let's say ovary. If any one of you has helped your mother taking out the seeds of peas, have you done it? So the seeds are ovule, right? Ripened ones. So do you remember when you used to, this is the seed, the pod. In the pod, there was something like this, and there is the seed. Something like this, and there is the seed. Something like this, and there is the seed, right? So when you take the seed out, this thing stays back. Remember doing that, people? How many of you have done it? That was my first employment. I used to get 5 rupees to do this. My mother employed me to do this whenever we used to make anything with peas. Great. So you people know what I'm talking about. So this wall that you're opening, the pod, is basically the ovarian wall. And inside it, what you are taking out is a ripened ovule, a developed ovule, which has become seed. And what you are leaving behind is the placenta and the funicle. Okay. So placenta, funicle, and at the junction where it is fused with the body of the ovule here is called hilum. Okay, hilum. So write down. Hilum represents the junction between ovule and funicle. Hilum represents the junction between ovule and funicle. And there is one question on hilum that comes in, I've seen in many entrances. So hilum in plants is analogous. Can you think of what it is, what it can be analogous to in an animal, in a human? Anyone? Think about it. I just want anyone to just give an answer. Think of a human, the reproduction, sexual reproduction happening in human. What it is. So hilum, let me give you a hint. What happens to that pea seed that you have plugged from the pod? Did you ever looked at that pea seed after you plugged? You're like, okay, who pays so much attention? I never looked, so I'll not say that, I was so observant. But if any one of you did, this is a pea seed. Okay, let's make it a little cute. This is a cute pea seed, and you broke it from the pod. So do you see a scar left from where it got detached from the pod? Yeah. And now think of a cute human baby. I won't be able to make a very cute baby. Oh God, this is atrocious, a baby. But when the baby gets plucked out of the mother's uterus, do you see a scar that's left? Yeah. Forget about the hands. It looks like flipper of a whale, but so the hilum is known as belly button of the seed. So the hilum, the junction point, is an analogous structure to the navel where the umbilical cord is attached to a human, right? So if you imagine a human baby as a seed, which is attached through something to placenta, right? So here the seed, basically this part is the seed, and it is attached to placenta through this funicle, through this common junction called hilum. So when you pluck it, it remains as a scar on the seed. Okay. Similarly, like when a human is born, there is a scar in our bellies, which is the navel or the belly button. That actually is a regression of umbilical cord. When we were inside, it was the umbilical cord. So write down, hilum is an analogous structure. Hilum in plants is analogous structure to umbilical cord and belly button in humans. Both leave scars, okay? It's also called the belly button. It leaves a scar, which is called belly button of seeds. This information, I'm not sure if it is an NCRT, but I have seen questions on this. Yeah, Iram reminds me I should keep water bottle on my table. Thank you, Iram. I normally do, I forgot it today. Anyways, cool. So now let's go forward. So this is hilum and junction between ovule and funicle. Now each ovule has a couple of protective layers called integuments. Okay, so remember these layers that I'm drawing here. This one is called outer integument. Integument. This is called inner integument. So there can be a couple of layers just to protect the embryo sac. So what you see here in, let me give it a red color, very important. This is the embryo sac, which is at the center, at the heart of the ovule, because that's where everything important takes place. Okay. And around the embryo sac, no, before you go to embryo sac, around the embryo sac, the immediate tissues, which I'm trying to draw, like these cells, which just cover the embryo sac. See, around anything which is important, which needs to develop, the cells which will surround it will do two things, either protection or nutrition, right? So this region is called the cells of nucellus. Nucellus. Okay, so around the embryo sac is nucellus, and that nucellus is covered by integuments. Okay, so in the center is the embryo sac, it's covered by nucellus, and imagine it as a 3D from all sides, and that's covered by inner and outer integument, and these integuments connect to funicle through hilum, and then funicle is connected to placenta in the ovarian wall. Everything is clear till here, people? Yeah. Yes or no? No. Perfect. Now this tip that you see where there is a, this part where there is a opening like structure here is called a micropyle. Micropyle. Okay. It is a small opening. Micropyle simply means, micro means small, pile means the opening. So it's small opening where the integuments are absent. You can write down integuments are absent at the tip of the ovule called micropyle. And that end is called micropylar end, where micropyle is present. And now let's connect two things together. From the top, pollen was making a tube. Okay, like our protagonist hero of Shawshank Redemption, he's making a tunnel. And I told you that this tunnel does not enters into the embryo sac from top, it enters from the bottom. So where do you think the micropylar end is? At the top or at the bottom?
Bottom.
Bottom. Makes sense, right? Wherever there is a micropylar end, there is a hole, an opening, and through that opening, the tunnel can very easily go inside the embryo sac. So this embryo sac here, so the orientation in which I have drawn this is ulta, is reversed here, right? So if I ask you to take this and place it in this diagram, you will place it upside down. So the micropylar end should come here, and opposite to the micropylar end, this end, just for reference, it's called the chalaza. You know, I used to read it chalaja. I don't know for what reason. So chalaza or chalaza land it represents the basal part. Okay, write down. Chalazal end is the basal part of the ovule. From the basal part, from the chalazal end, pollen tubes cannot enter. They have to enter through the micropylar end. Okay. And now, remember, let's come to this diagram. Let me zoom into this. Yes. Let me zoom even further. But there's an art like this. People draw something, then zoom and draw something inside it, and then zoom, then keep drawing. I saw it on, I think, Instagram. So here, the hilum will be here, and the micropyle will be here, right? And this will be the chalazal end. If it has multiple ovules and one ovule is here, then it will be like this: chalazal end here and micropyle here. So to penetrate this one, the tube will have to cover even more distance and enter from here. And if there is a third one, funicle, micropyle, integuments, sorry, funicle, hilum, integuments, and this is the micropylar end. So for this, the tube will enter from here, right? You understood how it is attached? Because this is the wall of ovary. This is the wall of the sac. Clear, everyone? Give me a thumbs up. Well, it looks like art though. Perfect. Uh-oh, what happened? Nothing happened. Okay, all good so far. So everyone understood micropylar end, funicle, hilum, nucellus, embryo sac, integuments, chalaza, and chalazal end. Everything is clear? Now, about the process of megasporogenesis. Now, we understood microsporangium, we understood microsporogenesis, we understood pollen tube coming in, we understood pollen pistil interaction, we understood the megasporangium. For megasporogenesis, the process, let's define megasporogenesis. Write down. Okay, so let's do one thing. I think it's praying time for some of you at your respective places, and it's also where we should end the class. So